This is the immune system part 1, the evolution of the immune system. So when we talk about general immunity, the human body has trillions of bacteria living on or within it, and the job of that bacteria is to protect the body. We have a lot of healthy bacteria on our skin. That bacteria and other physical barriers prevent pathogens and foreign materials for getting into our other systems. We call this the innate immune system. It is the most basic first line of defense. Basically what it does is it activates a variety of immune cells that protect and stop pathogens and other molecules that cause diseases, and it creates a physical barrier. The adaptive immune system is the other type of immune system. When we talk about the innate immune system, it's immediate and it's utilized by all sorts of organisms. It is very general. In contrast, the adaptive immune system produces antibodies that are very specific to a certain pathogen or foreign particle, and we activate things called B and T cells or B and T lymphocytes to attack those specific things that they are made to attack. It is slower, it takes a little bit longer to respond, and it takes a few days before it might be effective, and we found that the adaptive immune system is only present in jawed vertebrates. When we're looking at the immune system, we have to think about the idea of homeostasis, because this is why we have an immune system. Our body needs to maintain a certain balance of temperature and nutrients and bacteria and all sorts of things, and these defense mechanisms were evolved to get rid of pathogens and return an organism back to its original state. When we look back at the very beginning of the immune system, or when organisms had an immune system, we see that ancestral amoeba, so like very basic organisms, performed phagocytosis to obtain food. It would be advantageous for them to be able to distinguish between self and non-self when ingesting other cells. So what that means is that they don't have any sense of what they are eating, so they could be eating another amoeba or their own self, and so they need to be able to distinguish between self and non-self. And so they basically developed receptors that allowed them to determine this distinction, which is truly what the immune system is, right? That I can tell what is self, right? What are my own cells and what is not mine, and how to get rid of the things that are not me. In the immune system, we see an example of cell communication, and so this allows us to kind of cover cell communication within a unit that we need to learn in AP Bio. The proposed cell communication mechanism that that ancestral amoeba may have used was a 3-part communication system. Truly all it was the reception of a signal molecule, transduction, which is basically all of these molecules relaying some sort of information, and then the response, what the cell does in response to that signal molecule. There are animations of this on YouTube. I would recommend looking one up. When we talk about cell communication now in the immune system, it is far more complex than just these 3 steps. There are different types of receptors with signature molecules. We call them PAMPs. Those are the signature molecules. And so take a minute, look at this, kind of glean what you can from it. Pause yourself here. What you should be able to determine is that not only on the outside of the cell, but also at the nucleus do we see that there are different types of receptors with different molecules. What we also see is that when a signal molecule binds to a helper protein, it starts an inflammatory response, which then triggers these different receptors into doing something, a cell response. These are 3 types of cell communication pathways. The first is in plants, the second is in Drosophila, which is a fruit fly, and the third is in mammals. Take a moment to look at any similarities and any differences, and think about why they probably evolved in this way. Plants being the most basic, mammals being the most complicated. So when we talk about pretty basic mechanisms, we look at the idea of plants, right, which are our predecessors ancestrally. They have a lot of physical barriers, the epidermis, thorns, hair, spines. They have something called secondary metabolites, which are going to be enzymes that basically kill or make the herbivores ill. These are like proteins in the plant that they cannot ingest, and so then they do not get eaten. And then they actually have an immune response, but it is localized apoptosis, which basically means that we're going to see cell death and like withering on certain parts of the plants. This is why if there is a certain area of a plant that is totally brown, something probably happened to that area and it caused itself to die and like shut off that part of the plant so that the disease didn't spread. These are some other examples of the physical defenses and why we have them. I don't need to go into it too much. That's why we see these physical defenses. These are going to be your secondary metabolites. These are two examples of secondary metabolites, cannabinine and arginine. These are two very similar metabolites, two very similar compounds. One of them, just a small change, will create a protein that is poisonous for whomever is ingesting it. These are other secondary metabolites. You don't need to know these. I would understand what a secondary metabolite is and be able to give some examples of plants that might have them. So look on the far right, but you don't need to know the specific metabolites' names. We see that plants have a very specific defense, and then as organisms, as animals start to evolve, we see coevolution, that as one plant develops some sort of secondary metabolite, the animal starts to develop a way to eat that metabolite. So for example, this butterfly can break down a secondary metabolite that's produced by what we call the passion vine, and it uses it in protein metabolism, so the digestion of proteins. And so we see that there's coevolution. The plant evolved first, creating this metabolite to prevent it from being eaten, but then we see that the moth evolves alongside it. When we talk about the final step of the plant immune system, the induced defense response, certain pathogens that show up will produce molecules that produce a hypersensitive response. And so depending on the pathogen, if they show up, the plant will start creating molecules that create a hypersensitive response. The hypersensitive response is essentially the localized death of a plant and the plant cells very quickly, which is why we see these brown spots. That area died to try to prevent the spread of whatever pathogen we saw. What we'll also see is that a plant can have systemic acquired resistance, which allows it to become resistant to whatever pathogen we are dealing with. Methylsalicylate is what is released during the hypersensitive response while things are dying. Once that's converted to salicylic acid, it activates the SAR, or the acquired resistance, that keeps the plant healthy for a certain amount of days. And we're back. So we see that it only lasts for a certain amount of time, so if the disease is persistent, then that's when we see problems. So moving on to invertebrates and immunity. So we've talked about plants, we talked about amoeba, we're now going to talk about invertebrates. The innate immunity of a fruit fly, we see that it has an exoskeleton made of chitin or chitin. It is a protein similar to what's in your nails and in your hair. It also lines the digestive tract. The other thing that these organisms have is an enzyme called lysozyme, which is found in the gut of the cell walls, and it breaks down bacteria. In an invertebrate, they also have innate immunity. If a pathogen escapes the digestive tract, so it gets through the membrane, it will be in a fluid called hemolymph. Moving throughout this hemolymph, this fluid around the digestive system, are cells we call hemocytes, which destroy the pathogen and then eat it through a process called phagocytosis, and they'll digest it and get rid of the waste. Some hemocytes have toll receptors that produce antimicrobial peptides, which inactivate or kill fungal or bacteria by disrupting the plasma membranes. And so what we talk about with toll receptors is that we actually see like a inflammatory response. Each toll receptor identifies certain molecules as a pathogen or a foreign molecule, so you have to have a certain disease to have those toll receptors set off. So this idea of a toll receptor is that it recognizes a very specific signature molecule. It activates a signal transduction pathway, often producing what we call transcription factors, which when we talk about DNA and RNA, we should know that transcription factors are things that allow us to transcribe DNA, which is part of making proteins for a certain response. These transcription factors activate genes that are needed to synthesize antimicrobial peptides, or proteins, for destroying the pathogens. And so truly, the purpose of a toll receptor is to activate the creation of proteins to destroy the pathogen. And to make a protein, we know that we have to read through the DNA, make RNA, and create our proteins. When we wanted to talk about, or when people started researching the evidence for how this happened, they looked at fruit flies that were engineered with a fluorescent protein, and they were stabbed with a sterile needle. They were also given bacteria, and they were able to track essentially what happened in the fly.